The relationship between genotype and phenotype is rarely simple. So genotype is the actual alleles that you have for the genes. So for example, big A, little a, or whatever. And the phenotype would be the physical stuff. So what do you actually look like? What's your blood type? That kind of thing. Mendel had a good fortune, or chose well, to choose a system that was relatively simple genetically. Traits that were controlled by a single gene and genes that had only two alleles each. And they each showed complete dominance. So he looked at the color of the peas and the color of the pea pods and the shape of the peas, that kind of thing. And they all were fairly simple, with simple dominance and with just two alleles per gene. But in the real world, most traits are actually controlled by many genes. So there are more complex interactions, and some examples are pleiotropy, polygenic inheritance, which includes epistasis, and environmental factors. So let's take a look at each of these. So the first one is pleiotropy. Pleitrophy occurs when a single gene influences many traits. So therefore, a new mutation in the gene might have an effect on many traits. So for example, let's talk about albinism. That could be one mutation, and it affects the color of your hair, the color of your skin, your ability to see, which you might not think is super related, but there's melanin on the inside of your eye, just under the retina. So here's your eyeball, and it will certainly affect the color of the iris, which is this part, but light will go through here, and it'll go to the retina, and that's what detects light. But right on this side of the retina is the choroid coat, and that's got melanin in it, which will absorb light. So that way, light is detected, and then it gets absorbed right away. It doesn't go bouncing around all over the place in the eye, causing lots of problems. So anyway, that one mutation in how to make melanin can affect lots of diverse things, so from your skin color to your vision. So the underlying mechanism is that the gene codes for a protein that's used by many different cells or that can signal many cell types. So some mutations just affect one thing, but some mutations affect a ton of things. So another example of pleiotropy is the human disease PKU, phenylketonuria. It's caused by a mutation in a single gene that codes for an enzyme. The enzyme is phenylalanine hydroxylase, and it converts the amino acid phenylalanine to tyrosine, which is another amino acid. So this one mutation can cause all kinds of things, because if you don't convert this amino acid, it builds up. It can cause mental retardation, reduced hair, and skin pigmentation. So it causes diverse effects. Here's another example, polygenic inheritance, an example of complexity. Polygenic is just what it sounds like, many genes. So for example, your skin color is coded for by more than one gene, as is your eye color. So the inheritance of a trait that is caused by two or more genes. So something like widow's peak is caused by one gene, but something like skin color is caused by many. So new definition, locus. This is the specific location of a particular gene on a chromosome. So different genes have different loci, but alleles for a gene are found at the same locus. So in other words, here's chromosome 15. And I could have it backwards, but let's go with it. Here's the gene for whether you're going to have brown eyes or blue eyes. So you have two alleles at this one locus. And that's chromosome 15. Chromosome pair 15. Here's chromosome pair 19. Totally different location, right? We're pretending I'm drawing homologous chromosomes even though they don't look like it. Totally different chromosome, different DNA, totally different gene, but codes for the same thing, eye color. So this is a different locus, and these are two alleles at this locus, these are two alleles at this locus. Both affect what color eyes you have, whether they're going to be brown or blue, and whether they're going to be green or blue. So the iris, this part. So polygenic traits do not follow simple Mendelian patterns of inheritance. Instead, their phenotypes typically vary along a continuous gradient. So for example, with with dogs, Labrador retrievers, they can be blonde or black or brown, and that's kind of it. Whereas with people, there's a blended gradient for color. So people can be from very, very light to very, very dark with everything in between. So an example of a polygenic trait is human skin color. Several genes factor into determining a person's natural skin color. So modifying only one of these genes changes the color only slightly. So imagine that each gene has two alleles, one light and one dark, and imagine that they demonstrate incomplete dominance. So, and let's say there's like 7 genes for it, but we're just going to look at 3 genes because it's easier. So dominant AA, dominant BB, dominant CC would be someone very dark, and little a, little a, little b, little c is someone very light. Imagine that there's not 3, but 7 or 8 or 9 genes for this, and it gets a little bit more complicated. But anyway, just looking at 3 possible alleles. Here's A, B, C, and here's A, B, C. And so these are 2 people with kind of medium skin color. If you imagine their skin color is the same color as the thing right here, you can imagine that this one could make an egg that's little a, little b, and little c, right, randomly. And that one can make a sperm that's little a, little b, and little c. If that sperm fertilizes that egg, then the baby could be very light color. Of course, this person can also make an egg that has these genes. A, B, and C. And this person can make a sperm that's A, B, and C. So if that fertilizes that, you get someone who's very dark. So these people of medium color could end up making a baby that's very light and a baby that's very dark. Now, most of the time, they're going to be somewhere in between, right? This one will make an egg that's like A, B, C, and that one will make an egg like this, A, B, C, something like that. And so they're going to end up mainly with babies with medium skin color. So that's what this bell curve is showing you, that they have a huge chance of having babies that are kind of the same color they are, more or less. But there's this teeny little chance that the babies could be very light or very dark. And of course, this is only true for people who are heterozygous for these genes. So a cross between 2 heterozygous individuals, heterozygous for everything, they would have an intermediate skin shade, and they could produce offspring covering a wide variety of shades, from very, very light to fairly dark. Individuals with intermediate skin shades would be the most likely offspring, that's why the bell curve is high here, but very light and very dark individuals are possible as well. Oops. Epistasis. Sorry about that. So epistasis is a gene at one locus, and it alters the phenotypic expression of a gene at a second locus. And sometimes you get these unexpected phenotypes that occur, and you have expected ones that don't occur. So it doesn't make sense until I show you an example. So let's look at this. So for example, in mice and many other mammals, fur color depends on 2 genes. One determines whether the pigment will be deposited in hair or not. So big C means you'll have some color, little c means you won't, so you'll be very light. And then a second gene determines whether the pigment's going to be brown, black or brown. So the black allele is dominant to the brown allele. So here are a bunch of mice. Kind of cute, huh? Believe it or not. An individual that is little c has white, so they're albino, regardless of the genotype of the second gene. So these 3, doesn't really matter what their B's are. These are all albino. This one is going to be brown, like that. And these are both going to be black. So black, black, brown, and all the others. Doesn't matter what this is, because the C is more important. These are all albino, white. So a cross between 2 black mice that are heterozygous for both traits will follow the law of independent assortment. So big B, big C, little c. Those are going to be black because the big C says they have color and the big B says they're black. So their gametes would be big B, big C, right? You foil it first. And then big B, little c. And I'm going to be really careful here to make my little c's look different from my big C's. First, outer, inner, little b, big C. And finally, little b, little c. And so those are the gametes, and both of them would have these gametes. So let's look at that. So both of them can make a big B, big C, then a little b, big C, big B, little c, and little b, big C. So these 2 black mice you would think it wouldn't be like this, but they can actually make a bunch of black babies, a couple of brown babies, or some white babies. So it's a little bit different from what we've seen before. So unlike the 9 to 3 to 3 to 1 offspring ratio of a normal Mendelian experiment, the ratio is 9 black to 3 brown to 4 white. So you're kind of combining these. A similar thing happens in dogs. A brown-haired male whose father was blonde, so blonde is yellow, but it's like the albino from the last, at least genetically from the last choice. So a brown-haired male whose father was blonde. Now, if this brown-haired male is brown, it means he's little b, little b. He has to have at least one big C or else he'd be blonde. And he has to have a little c because his father was blonde. He's mated with a black-haired female whose mother, oh, and the black-haired female has to have a big C, right? Whose mother was brown, whose father was blonde. Whoa. And so we want to know what happens there. So this one makes the gametes little b, big C, and little b, little c. And this one makes the gametes big B, big C, big B, little c. Let's see, little b, sorry, big C, and little b, little c. So if we did, I think the next page might have the whole thing done up. So what's the expected ratios of their offspring? So let's take a look at that. So if you do the whole pun, you could do these twice if you want to, but this is just as good. This one is black. Big B, big C, that's black. That one's black. And that's it. This one is blonde. Let's see, that one's blonde. So 3 black, 2 blonde. They're blonde because they're little C's. Let's see, that one's brown, and that one's brown. So 3 brown. So those are the ratios you'd expect in their kids. Yeah, did I say that too blonde? Hopefully. Okay, here's another example in chickens. So in other cases of epistasis, interaction between the 2 gene pairs results in unexpected, in a phenotype that neither pair can produce alone. So this has to do with their, I don't know, what do you call those things? They're combs. So a single comb is little r, little r, little p, little p. P comb looks like this, and it's at least one big P with little r's. And then this kind of funny looking one has a big R with whatever, either another big R or a little r. Little p's. And then if you have big R and big P, you get this walnut comb one. And so you get this unexpected different result. It's not that one is telling the other one not to happen, it's that you get this brand new thing happening. So, all right, that's just the summary of the whole thing, so I should kind of explain this a little bit. So a single comb mated with a single comb will all make single combs. But you could have a rose comb one, so big R, little p, and mate that with, let's say, a P comb, little r, big P, little p. And so what you're going to get with that is this one will make big R, little p, and little r, little p. And this one can make little r, big P, and little r, little p. And so this one would be a single comb because it would be little r, little p, little p. And this one would be big R and big P. So that would be a P comb. And then this one would have little r, big P, little p. So that would be a P comb also. This one would be big R, little p, little p. So that would be a rose comb. The last thing that I wanted to mention is that phenotype depends on the environment too, as well as genes. So for example, a single tree has leaves that vary in size and shape, how green they are, and it all depends on wind and sun. So there's more than genetics typically. So for humans, nutrition influences height along with genes. Exercise will alter build along with genes. Sun tanning will darken the skin along with genes. And then experience can also improve your test, your performance on intelligence tests. So study, study, study. So the product of a genotype is sometimes very clear. Like, for example, if you end up with type O blood, there's really nothing you're going to do about that. Sometimes not so clear. So in most cases, though, especially polygenic traits, there's a range of phenotypic possibilities that are determined by the environment. So these are hydrangeas, one of my favorite flowers. They can be red or purple. I think the purple happens with acidic soil and the red with basic soil. So it depends on both their genes and their environment. Finally, linked genes. Genetic linkage occurs when particular alleles are inherited together, and genes that are on the same chromosome are more likely to be inherited together and are said to be linked. So linked genes usually do not sort independently because they are located on the same chromosome and tend to move together through meiosis and fertilization. So for example, this gene and this gene are very close together. Usually they're going to be inherited together. This gene and this gene are very far apart, so chances are good that there'll be a crossover, so they're typically not inherited together. So the frequency of crossover is greater the further apart two genes are, and this is proportional to the relative distance between the two genes. So you can actually calculate out how close genes are based on how many times they cross over to make different offspring types, different gamete types. And that's the end of my story.